Hey fellow lab rats, this is Rebecca from the Lab Rat YouTube channel. In this video, I'm going to be discussing enzyme histochemistry and its uses in the histology laboratory. All right, let's get started. So let's first talk about enzymes. So these are proteins that help catalyze or speed up chemical reactions without being consumed or permanently altered by that chemical reaction. Enzymes are found in every cell and organ in the human body and are responsible for many essential functions, including the clotting of blood and also digestion. Enzymes bind to a specific substrate molecule at specific locations called active sites. The active site is shaped in a way that only certain substrates can fit into, and this is commonly referred to as a lock and key. A cofactor is a non-protein chemical compound or metallic ion that is required for some enzymes' role as a catalyst. So these are considered helper molecules for that enzyme. Enzymes catalyze oxidation and reduction reactions, also called redox reactions. Oxidase is an enzyme that can catalyze the oxidation of substances by the addition of oxygen, and dehydrogenase can catalyze the removal of hydrogen from material molecules. Oxidation can also occur with the loss of electrons. Reduction can occur with the loss of oxygen or the gain of hydrogen or electrons. Even a moderate amount of heat can inactivate enzymes, and this is an irreversible alteration of that enzyme. Enzymes are also sensitive to pH changes, so some enzymes work the best at an alkaline pH, which is a basic pH, and there are those that work optimally at an acidic pH. Most, however, work best at a pH that is close to neutral, so around 7.0. Enzymes recognize its corresponding substrate by its chemical groups. So these are factors that influence the demonstration of enzymes within tissue. The treatment of tissue before and during the procedure. So a lot of enzymes are destroyed by the fixation process. Some are sensitive to freezing and thawing steps. And because of this, compromises must be made. So one may sacrifice some activity for better localization of the enzyme, or they sacrifice the enzyme diffusion for maintaining maximum activity of that enzyme. Also, sometimes optimal substrate concentration cannot be obtained. This may be caused by poor solubility of the substrate, or the substrate may be hydrolyzed or broken down more slowly than other available substrates. So we've discussed before that there's an optimal temperature for enzyme activity. So if the temperature is not optimal, this of course can affect the demonstration of enzymes. So enzyme activity is usually destroyed at temperatures that are greater than 56 degrees Celsius. A non-optimal pH can also affect the demonstration of enzymes. So of course there's an optimal pH for each enzyme, and most of them function the best at a neutral pH, so a pH of around 7.0. There are some azodiazine-simultaneous coupling methods that use a non-optimal pH, which lowers the activity of enzymes. Also, there can be inhibitors, so enzyme inhibitors. So inhibitors are molecules that bind to enzymes and reduce or prevent the activity of that enzyme. Examples of these are an excess of diazonium salts in the substrate, fixatives, heat, and some metallic ions. These can decrease or completely prevent the activity of the enzyme. Enzymes are specific to the type of reaction that they catalyze. Enzymes are typically named to that specific substrate or molecule on which they act, with the ending of ACE, so ASE at the end of them. Some older enzymes are not named this way, so these are examples of this would be trypsin or pepsin. So trypsin is an enzyme that helps in the digestion by breaking down proteins within the small intestine. And pepsin is also an enzyme that helps in digestion, specifically by breaking down proteins into amino acids and smaller peptides. So those don't end in ACE, but all the other enzymes do. There are 6 basic reaction classifications of enzymes. Oxidoreductases, transferases, hydrolases, lysases, isomerases, and ligases. Hydrolases are a type of enzyme that act on various substances, usually through the addition of water. In some cases, however, water can be removed in these reactions. There are 3 broad subclasses of hydrolases: esterases, phosphatases, and peptidases. Esterases are a hydrolase that catalyze the hydrolysis of ester linkages. Phosphatases are hydrolases that hydrolyze esters of phosphoric acid. And peptidases are hydrolases that attack peptide bonds. So we'll talk more about these first 2 here individually in just a bit. So hydrolases. So there are 4 types of histochemical reactions that are used for the demonstration of hydrolytic enzymes. The first is by simultaneous capture or coupling. So this is the most common technique used for the demonstration of hydrolytic enzymes. So this reaction involves the release of a reaction product by the action of the enzyme on its substrate. That reaction product is captured or coupled either with a diazonium salt to give an insoluble azo dye or with a metallic ion. In the reaction that uses the diazonium salt, both the diazonium compound and the substrate are present within the incubating medium. In the metallic ion precipitation methods of Gomori, the initial coupled reaction product is usually not visible. So the metal substitution methods are used to produce a visible reaction product. The second reaction type is post-incubation coupling. This reaction depends on the formation of an insoluble reaction product, which must remain at the reaction site for the entire incubation period. The sections are then treated with a coupler and a different separate step. The third reaction type is self-colored substrate. So in this technique, the reaction product must be both insoluble and colored. It uses a soluble colored substrate and no coupler or capture agent is necessary. The last reaction type is intermolecular rearrangement. In this reaction, the substrate is soluble and hydrolysis causes a rearrangement of the molecules, which results in an insoluble color reaction product. Okay, so remember how I said previously that there are 3 classes of hydrolases: esterases, phosphatases, and peptidases. So the esterases are enzymes that are capable of breaking the bond between carboxylic acid and alcohols, phenols, or naphthols. They are further divided into classifications: specific and non-specific esterases, although the classifications are not very straightforward, unfortunately. There is overlap in substrate specificity and concentration, optimal pH ranges, and their response to activators and inhibitors. But a majority of both specific and non-specific esterases are capable of hydrolyzing alpha-naphthol acetate. A diazonium compound is used in the substrate for the purpose of being a coupling reagent. So this results in the formation of an insoluble azo dye. The most rapid coupling in this reaction occurs when the pH is from 6 to 6.5. Now, phosphatases are another classification of hydrolase. So these are hydrolytic enzymes that break the bond between an alcohol and a phosphatase group. This enzyme type is present in a lot of plants and animal tissues. Some phosphatases act specifically on a single substrate. So an example of this is ATPase. So ATPase is found in the mitochondria, and it is actually bound to myosin, which is a contractile muscle protein. Now, other phosphatases act with less specificity to their substrates, and these are divided into 2 groups: those that have optimal activity at an alkaline pH, called alkaline phosphatases, and those that have optimal activity at a low pH, called acid phosphatases. Both acid and alkaline phosphatases are present in lysosomes. Oxidoreductases are another classification of enzyme. These include oxidases, peroxidases, and dehydrogenases. Oxidase use molecular oxygen as the hydrogen acceptor, resulting in the formation of water. Peroxidases catalyze the oxidation of substrates by hydrogen peroxide, and dehydrogenases remove hydrogen atoms from an organic substrate. Dehydrogenases usually transfer those hydrogen atoms to a coenzyme, and then from that coenzyme, that hydrogen is then transferred to acceptors called the electron transport system. This system is very important in the production of cellular energy through coupling with oxidative phosphorylation. The final acceptor for those hydrogen atoms in that process is molecular oxygen, which results in the formation of water. So I said that the oxidoreductases are oxidases, peroxidases, and dehydrogenases. Diaphorases are a type of dehydrogenase. So most dehydrogenases are associated with a coenzyme that functions as a hydrogen and electron acceptor. Usually that coenzyme is nicotinamide adenine dinucleotide, or NAD for short, or nicotinamide adenine dinucleotide phosphate, or NADP for short. As NAD and NADP accept the electrons in the reaction, they become NADH or NADPH. Diaphorases are dehydrogenases with the sole purpose of oxidizing that NADH and NADPH. Diaphorases are flavoproteins that contain flavine adenine dinucleotide. So they are also called tetrazolium reductases. These are firmly bound in mitochondria and the endoplasmic reticulum. They are really important in muscle enzyme histochemistry. It's an enzyme that is commonly used as a marker for the presence or the detection of mitochondria. Transferases are another type of enzyme. They're responsible for transferring a functional group from one compound to another. There are a lot of transferases, but phosphorylase is the only one we'll talk about in histology. Phosphorylases are transferase enzymes that are responsible for transferring phosphate groups. They are present in both animal and plant tissues and catalyze many reversible reactions that lead to the production and decomposition of saccharides. And saccharides are sugars. In vivo, meaning within the body, the phosphorylase is present in the cytoplasm and functions only in the degradation or breakdown of glycogen. In vitro, muscle phosphorylase enzymes catalyze the production and hydrolysis of an unbranched polysaccharide of amylose type. The length of the unbranched chain is proportional to the amount of phosphorylase activity present. So when stained with iodine, the color varies depending on the length of the chain. So a negative reaction will be yellow. And then varying shades of brown, lavender, purple, and intense blue-black are seen with increasing chain length. In an ideal situation, tissue that is used for the demonstration or detection of enzymes should be fixed. It should be fixed before freezing if possible, as diffusion artifact may be seen in unfixed frozen sections. Also, post-fixation of sections will not prevent diffusion. Certain soluble enzymes require some fixation before the freezing of the sample, or an insufficient amount of enzyme will be retained for demonstration. Some enzymes will be inactivated by even the smallest amount of fixation. Cold formalin, so formalin around 3 to 4 degrees Celsius, cold calcium formalin, and cold acetone can be used to fix tissue for some enzyme studies. After the tissue has been fixed, it can be stored in gum sucrose solution at 4 degrees Celsius for several weeks. If the technique being used permits the fixation of the sample, blood smears should be fixed immediately once they are dried. If not immediately fixed, the enzyme activity will be decreased in the sample. Muscle biopsies are not fixed and are just frozen, so whenever possible, sections are then post-fixed, meaning they are fixed after they've been frozen. Muscle tissue is really prone to ice crystal artifact, so the cryostat freezer plate is used for freezing tissue, but for muscle specimens, a more rapid freezing technique is recommended. Isopentane suspended in liquid nitrogen is a preferred method of freezing for muscle tissue. The isopentane must reach a temperature of -150 degrees Celsius before the muscle specimen is placed in it. If just liquid nitrogen must be used alone for freezing, the muscle should be dusted with talc, and talc is a mineral powder. If talc is not used and the specimen is put into the liquid nitrogen without it, some gaseous nitrogen will surround the specimen and the freezing will be impeded. In 1861, the first enzyme was used for the digestion of tissues. Beale used gastric juice to digest excess tissue from nerve fibers. Later, in 1868, Klebs, and in 1872, Struve began histochemical demonstration of enzymes. Specifically, they demonstrated that guaiac gives a blue color when applied to pus. This was actually the first demonstration of peroxidase. Guaiac is a resin that comes from a specific species of trees, and peroxidase is an enzyme that breaks down peroxide. In 1900, Brandenburg demonstrated the presence of peroxidase in the granules of leukocytes, and leukocytes are white blood cells, and some, specifically granulocytes, contain granules. The primary use of enzyme histochemistry nowadays is for the diagnosis of disease within the muscle. Some of the same techniques can also be used in hematology or hematopathology for the differentiation of leukemias. Leukemia is a broad term for cancers of the blood cells. The type of leukemia depends on the type of blood cell that becomes cancer and also if it grows quickly or slowly. Because most of the methods in enzyme histochemistry are for the detection of muscle disease, it's important to discuss and understand the histology of muscles. Individual skeletal muscle fibers or cells are usually around 30 to 80 micrometers in diameter and can be as long as 35 centimeters in length. Skeletal muscle fibers contain multiple elongated nuclei that are located at regular intervals just beneath the cell membrane called the sarcolemma. They also contain regular cross striations that are from the arrangement of actin and myosin, which are contractile proteins. Every muscle fiber has myofibrils, which are cylindrical structures with prominent cross striations. The myofibrils are parallel to the cell membrane or sarcolemma and with the cross striations. With the use of histochemical reactions, individual myofibrils can be identified. The term intrafibrillar space refers to the space between the myofibrils. So between the intrafibrillar space and beneath the sarcolemma is filled with the cytoplasm called the sarcoplasma. The sarcolemma is the cell membrane that surrounds the skeletal muscle fiber. And the sarcoplasm is the cytoplasm of the muscle fiber. Inside the sarcoplasm are mitochondria, intermediate filaments, free ribosomes, the Golgi apparatus, glycogen, and lipid droplets. Fascicles is the term of bundles of muscle fibers. These fascicles are surrounded by a dense layer of collagen called the paramecium. So the paramecium is the dense layer of collagen that surrounds the fascicles. The epimysium is a collagenous connective tissue sheath that surrounds the entire muscle. Individual muscle fibers are polygonal in shape and are pretty uniform in their diameter. These muscle fibers are separated from each other by a network of fine collagen fibers called the endomysium. The endomysium is not usually seen with the light microscope, but in some pathological conditions of the muscles can be very striking. Blood vessels and nerves are usually seen within both the paramysium or paramecium and the endomysium. So capillaries may also be noted in the endomysium. Muscles that are responsible for helping to maintain a body's posture are required to contract continuously, while others, like extraocular muscle, make rapid contractions of brief duration. So the extraocular muscles are the muscles of the eye. So in the human body, these types of muscles cannot be distinguished from each other based on the color of the muscle. And so when I'm talking about that, I'm talking about the ones that have to contract continuously and those that are like more rapid contractions, all right? Again, these type of muscles can't be distinguished from each other based on the color of the muscle in humans. So in some species, like in chickens, the two types are easily distinguished based on color. So thigh muscles are red and breast muscles are white in chickens. The slow twitch red muscle fibers are primarily aerobic or oxidative in metabolism. They have a lot of mitochondria, lipids, and myoglobin and have a very good blood supply to them. These type of muscle cells are called type 1 fibers. Type 2 fibers are fast twitch white fibers and have anaerobic metabolism. They only have a few mitochondria, little myoglobin, and have a pretty poor blood supply to them. These type 2 fibers are rich in glycogen and glycolytic enzymes and have a high resistance to fatigue. Now, type 2 muscle fibers are further divided from one another by type A, B, and C types. Type 2C muscle fibers are not normally seen in normal adult muscle because these fiber types are undifferentiated and precursors of other types. In humans, muscles contain roughly 33% type 1 fibers and 67% type 2 fibers, give or take. The type 2 fibers are divided into 50% type 2A fibers and 50% type 2B fibers. In normal human muscles, type 1, 2A, and 2B fibers are distributed in a checkerboard pattern. So here is a drawing showing muscle fibers. So this is a skeletal muscle fiber on top here. You can see the epimysium or epimysium here. So let's see here. This is what I'm talking about right here. Okay. You can, so recall that epimysium is the collagenous connective tissue sheath that surrounds the entire muscle. There are the muscle fascicles here. Okay, muscle fascicles. And those are bundles of muscle fibers. Now the middle drawing here, okay, shows a muscle fascicle, right? So the paramysium and the endomysium are visible here. So the paramysium or the paramecium is that dense layer of collagen that surrounds the fascicles, and the endomysium is the network of fine collagen fibers that separate the muscle fibers. So here, paramysium, endomysium, and this muscle, so this is the muscle fascicle, okay, and then inside here shows the muscle fascicle and then the individual muscle fiber here, all right. So the bottom drawing is the individual muscle fiber, okay, and this shows the sarcolemma, which is the membrane that surrounds the skeletal muscle fiber. So hopefully these cartoons or these drawings kind of give you an idea of how these things are arranged. So sometimes it's good to have pictures showing you what these terms are. So now that we've discussed the histology of muscles, pathological changes in the muscle can frequently be seen on the H&E stain. And H&E, remember, is hematoxylin eosin stain. So these pathological changes can be changes in the size, so atrophy or hypertrophy of both individual fibers and groups of fibers. So atrophy is the wasting or thinning of muscle mass, where hypertrophy is the increase of size of skeletal muscle through the growth of their cells. So there can also be changes in the location and the appearance of the muscle cell's nuclei. There can be inflammatory cells present or the presence of degenerating or regenerating muscle fibers. There can also be an increase in connective tissue. So the photo on the slide here shows a normal skeletal muscle section that is stained with the H&E stain. The fibers are pretty uniform in diameter and have peripherally located nuclei. Minimal freezing artifact can be seen here. So each of these, I'm going to use, let's use blue. So each of these, okay, are the muscle fibers, and they're all pretty uniform, right? Okay, so this is a normal, this is normal, okay? Histochemical techniques are required to demonstrate other changes such as type grouping, fiber type predominance, so what type of fibers are predominant in the sample, central cores, or target fibers. Using a panel of histochemical stains in conjunction with the H&E stains can help to diagnose most abnormal biopsy findings resulting from a primary muscle disease, which is called a myopathy, or secondary to denervation, which is a neuropathy. So this is a chart that comes from Dr. Carson's book that breaks down the normal histochemical reactions of muscle fibers. So I'm not going to talk about this in detail, but just know, so the enzyme technique is here, and then these are the different muscle fiber types and their reaction below. So make sure to know this. This will be helpful for you on your histology journey. So the photo on the left-hand side of the slide shows a frozen muscle section that's of course stained with the H&E stain. It shows a marked variation in muscle fiber size. There are also numerous fibers that have atrophy that are in small groups. So you see how there's variations between all of these, and like for example, these are all much smaller than these, right? Okay, so those are the fibers that are experiencing atrophy. So this section suggests the presence of a neuropathic disease or neuropathy. So the photo on the right hand side shows an H&E stain frozen section, again of muscle, and over here on this side here, those are normal muscle fibers, but you can see over here something very, very strange is happening, okay? So there's muscle fibers here, there's like 3 muscle fibers, okay, that are showing signs of phagocytosis. So this is actually a degenerative change that is seen in some common inflammatory myopathies. So this is, both of these are abnormal muscle sections. As I've stated previously, muscle tissue is extremely prone to ice crystal artifact. Unfortunately, this artifact can make the muscle biopsy stains unreadable. So it's very important to correctly freeze all muscle tissue. So the following is Frida Carson's recommended procedure for freezing muscle biopsy specimens. So the first step is to orient the fresh unfixed muscle biopsy tissue so that cross sections will be obtained. Write the patient's name and identifiers on the side of a piece of cork with a waterproof marker. You want to punch a hole in the edge of the cork and put a piece of thread through that hole. The tissue should then be mounted with a gum tragacanth. So this gum tragacanth will keep the muscle tissue standing up in the correct position. You don't want to use a whole lot, just enough to orient that muscle tissue properly on that cork. Then suspend a steel beaker of isopentane in a dewar flask that contains liquid nitrogen. The isopentane should reach a temperature of negative 150 degrees Celsius, and you must ensure this by using a thermometer. So this is a critical step in preventing ice crystal artifact. After the isopentane reaches this desired temperature, the cork with the muscle biopsy specimen should be placed into the isopentane for 15 seconds. After that 15-second mark, it should be put directly into liquid nitrogen. It can remain in the liquid nitrogen until it's time to cut it. The thread that was put through the hole in the cork can be taped to the outside of the liquid nitrogen container if it needs to stay in there, store until it's ready to be cut. After the liquid nitrogen step, the cork should be mounted on a disc with a freezing medium or water. There should be about a 10-minute warm-up for the tissue to reach the cryostat temperature, which is about negative 20 degrees Celsius. After sectioning the tissue, the cork should be removed from the disc, wrapped in plastic wrap, and then in foil. It should be labeled with tape and stored at negative 70 degrees Celsius. Place the wrapped specimen in liquid nitrogen to transport it to the freezer if it's far away from the work area. Sections should be collected in a slide box and stained as soon as possible. They can be kept in the freezer for several days if they are kept in a closed container. There may be a slight reduction in some enzyme activity when stored, but if stored properly, not too much. Let's look at some photos of this process. So this photo on the left-hand side of the slide here shows the small beaker with isopentane, okay, and it is lowered into the container of liquid nitrogen. The photo on the right hand side shows the muscle section that is oriented on the cork with the gum tragacanth, and then of course there's the thread, right? that's punched in. There's a hole in this cork board here, and that thread is put through that hole. Now the photo on the left-hand side shows the temperature of the isopentane being checked with the thermometer. Again, it must be at negative 150 degrees Celsius before the muscle is put into the liquid. The photo on the right-hand side shows the cork with muscle biopsy being submerged into the isopentane. Remember, the isopentane is in that metal container there, and then of course this is the liquid nitrogen in that container. Right? The left-hand side photo here shows the cork with the muscle biopsy attached to a metal cryostat disc for sectioning. The photo on the right shows a liquid nitrogen container. The beaker of isopentane can be suspended here in this container until it's ready to be cut. All right, so now we've gotten to the fun part here. So the stains that we use in histology, specifically for enzyme histochemistry. So the alpha-naphthyl acetate esterase stain is a non-specific esterase stain that is useful for differentiating between type 2 atrophy and neurogenic atrophy. So this is because denervated muscle fibers stain dark, and type 2 atrophy muscle fibers do not. All right, and denervated muscle fibers occur in that neurogenic atrophy. Motor end plates and lysosomes in inflammatory cells are also demonstrated with this stain. We've talked about esterases, and these are enzymes that hydrolyze ester bonds, and depending on their preference for substrates, they are classified as either specific or non-specific. Histiocytes, also called macrophages, are cells that play a role in the body's immune system and also of tissue repair. So histiocytes will stain with this method, as well as acetylcholinesterase accumulation and denervated fibers. The hydrolyzed alcoholic residue couples with the hexadized parerosinoline solution, which gives an insoluble brightly colored azo dye as the reaction product. So no fixative is needed for this stain. Frozen sections of unfixed tissue are cut at 10 microns and mounted on slides. For quality control, skeletal muscle tissue that contains inflammatory cells or motor end plates should be used. And I'm realizing I haven't talked about motor end plates, so forgive me. So motor end plates are also called neuromuscular junctions. So this is a chemical synapse that occurs that connects a motor neuron to a muscle fiber. So the reagents needed for the alpha-naphthyl acetate stain are as follows: 0.2 normal phosphate buffer with a pH of 7.2, parerosinoline stock solution, 4% sodium nitrite, 1 normal sodium hydroxide, and incubation solution. So the procedure for this stain is pretty simple and straightforward. The section should be incubated in incubating solution for 1 to 2 hours at 37 degrees Celsius in either a water bath or an incubator oven. After this time frame, the section should be washed in running water for 1 hour. After the 1 hour period, the section should be dehydrated through grated alcohols, cleared in xylene, and then mounted with a synthetic resin. When properly stained with the alpha-naphthyl acetate esterase stain, normal muscle fibers will be a very pale yellow. Motor end plates will be brick red in color. Denervated muscle fibers, macrophages, and lysosomes will be a dark red-brown color as well. The photo on the left-hand side of the slide shows a skeletal muscle section stained with the alpha-naphthyl acetate esterase stain. The very dark red-brown areas are motor end plates, so we're talking about here. Okay, those are motor end plates. The right-hand side photograph shows a skeletal muscle section stained with the same technique. The dark staining of the small angular fibers, so we're talking about here, here, some here, all right, are fibers that have been denervated, and that indicates that a neuropathic disease process has occurred. The naphthol ASD chloroacetate esterase technique is used to identify granulocytes in the classification of leukemias or in coloroma. So this is because esterase is found in lysosomes of some granulocytes. And granulocytes are white blood cells that have granules present in them. Coloroma is also called myeloid sarcoma, which is a rare cancerous tumor type. This is a specific esterase stain. It's unique in that it is one of the rare enzyme techniques that can be performed on paraffin-embedded tissue. Just like in the alpha-naphthyl acetate esterase technique, the hydrolyzed alcoholic residue couples with a hexazetized paracetylene to give brightly colored azodireaction product at the site of enzyme activity. If doing blood smears, it should be fixed for 2 minutes in absolute methanol formaldehyde. Any well-fixed tissue is acceptable except bone. Only EDTA decalcified bone is acceptable if you are using bone. Most aspirate or buffy coat smears have an internal quality control. For paraffin section, spleen tissue can be used as a quality control. So I use the term buffy coat. So this means a thin layer of white blood cells and platelets that separates the red blood cells from the plasma in a blood sample after the sample has been centrifuged. So remember, this can be used on blood or on tissue. Reagents for the naphthol ASD chloroacetate esterase technique are esterase solution A, B, and C, 0.1 normal HCl, 0.1 molar sodium barbital, working esterase solution, and Mayer hematoxylin. To perform the naphthol ASD chloroacetate esterase technique, tissues should be deparaffinized and hydrated. Stain and freshly prepared and filtered working esterase solution for 30 minutes. After 30 minutes, if the reaction is not completed, the solution should be refiltered and stained again for 30 minutes more. The slides should then be washed in running water for 5 minutes and then washed again in distilled water. Then, Mayer hematoxylin should be used for a counterstain for 5 minutes. After that 5 minutes in Mayer hematoxylin, the section should be washed in running water for 5 minutes to blue the nuclei. After that 5-minute running water wash, it then needs to be rinsed in distilled water. After the distilled water rinse, the section should be blotted completely dry, then air dried for several minutes. Once the sections are dried, dip in xylene and then mount with a synthetic resin. When stained properly with naphthol ASD chloroacetate esterase, positive cells, so granulocytes and mast cells, will be bright red in color. Nuclei will be blue and the background will be unstained. So this photo here is a paraffin section of cervix tissue that is stained with the naphthol ASD chloroacetate esterase stain. This shows a marked infiltrate of myelogenous leukemic cells, so this is seen in red. So let's use blue here. So all of this red here, okay, is what we're looking at, right? So those are an infiltrate of leukemic cells. So those are granulocytes. So this is a specific esterase stain that works well on paraffin-embedded tissue. And this is, of course, a paraffin-embedded cervix tissue. All right, so let's talk about the ATPase stain. So the purpose of the ATPase stain, which has pre-incubation at various pH levels, will differentiate type 1, type 2A, and type 2B fibers. And this, of course, helps to separate myopathic from neuropathic issues. Muscle ATPase is dependent on the influence of pH, which allows for subtyping of the fiber types. So this involves a Gamori-type metal precipitation of the hydrolyzed orthophosphoric acid with calcium. A series of reactions leads to the formation of a colored metallic sulfide. Frozen sections of unfixed tissue is cut at 10 microns and mounted on slides. You want to cut 3 slides, one labeled 4.3, the second labeled 4.6, and the third labeled 9.4. And these number labels indicate the pH level. Skeletal muscle contains an internal control for this stain. The reagents needed for the ATPase stain are barbital, acetate buffer stock solution, both A and B, sodium barbital solution, barbital acetate buffer working solution, incubating solution, 1% calcium chloride, 2% cobaltolus chloride, and ammonium sulfide solution. The procedure for the ATPase stain is as follows. So you want to prepare a working barbital acetate buffer solution at both pHs 4.6 and 4.3. So that is the working barbital acetate buffer solution. Then you want to make a sodium barbital solution. You want to adjust the pH of 20 milliliters of that sodium barbital solution to a pH of 10.4. So this serves as the alkaline pre-incubation, and 50 milliliters of sodium barbital solution to a pH of 9.4. Okay, so, and forgive me, I said 50, I meant 55. So 55 milliliters of that sodium barbital solution to a pH of 9.4. I misspoke, forgive me. So to prepare the incubation medium, you want to separate into 2 jars. You want to start slides of at the pH of 4.6 and 4.3 in solution 1A, and then leave in for 5 minutes. You then want to transfer to a jar of solution 1D at a pH of 9.4, use separate jars, and leave in for 3 minutes. You want to place the above slides together in the first jar of incubating medium, and then incubate at 30 minutes at a temperature of 37 degrees Celsius. 2 minutes into the incubation period, you want to start the 9.4 slides in solution 1E, which is at a pH of 10.4 for 14 minutes. Let's continue on the next slide. So next you want to place the pH 9.4 slides in the second jar of incubating medium for 14 minutes at 37 degrees Celsius. Then combine all the slides and rinse in 3 30-second changes of 1% calcium chloride. Then you want to place for 3 minutes in a cobaltolus chloride. Then wash the slides well in distilled water, about 2 changes of around 1 minute each. Then transfer to ammonium sulfide solution for 20 to 30 seconds, and you want to make sure you do this under the hood. After that period, that 20 to 30 second period, you want to wash in running water for 5 minutes, then dehydrate, clear, mount with synthetic resin. So I realize, especially when kind of explaining this procedure, that it's kind of complicated because there's different slides. So make sure when you're doing this to follow your laboratory's SOP, which is your standard operating procedures, and make sure to do it step by step. Everything will be labeled there for you. So again, the ATPase stain is for fiber typing. In normal muscle, approximately 1/3 of types 1, 2A, and 2B fibers are present. Rarely, type 2C fibers can be seen on the pH 4.3 slide stain intermediate between the dark type 1 and the light type 2 fibers. ATPase is found in the mitochondria of all cells or attached to myosin in the muscle. So ATPase in mitochondria is activated by magnesium and inhibited by calcium. So muscle ATPase is activated by calcium and inhibited by magnesium. So it's different in the mitochondria versus in the muscle. Calcium is an incubating solution and activates the muscle ATPase and inhibits it in the mitochondria. So when stained properly with the ATPase stain, on the pH of 9.4 slide, type 2 fibers are dark and type 1 fibers are light to unstained. On the pH 4.3 slide, type 1 fibers are dark and type 2 fibers are light to unstained. And on the pH 4.6 slide, type 1 fibers are dark, type 2A fibers are light, and type 2B fibers are intermediate. So it is important to know this. Let's look at some photos here. So the photo on the left-hand side is a frozen section of normal skeletal muscle stained for ATPase activity, and this is done at a pH of 4.6. So this is on the 4.6 slide. So this shows 3 distinct types of muscle fibers. Type 1 fibers are very dark, right? So these super dark ones, let's use blue, all right? This is what we're talking about, okay? So those type 1 fibers are very dark on the slide of ATPase slide of pH of 4.6. Type 2A fibers are very light. So these really light ones here, let's do these really light ones, all right? Okay. So that's the type 2A fibers, and the type 2B fibers are intermediate. So it's like these here. Okay. All right. So those are the type 2B fibers on a pH of 4.6. So this is a pH of 4.6 slide. Okay. All right. So again, normal muscle contains about 1/3 of each fiber type distributed in this like checkerboard pattern. So the photo on the right-hand side of the slide is a frozen section of muscle stained for ATPase at pH 9.4. So let's just write it down here. pH 9.4. Just as an FYI, I'm writing this with my mouse. That's why I look like I'm a first grader trying to draw and write things. My handwriting is actually pretty good, just not with a mouse. So this is a frozen section of muscle stain for ATPase at a pH of 9.4. So at this pH, type 1 fibers will be light, okay, and type 2 fibers will be dark. So note the small angular atrophy fibers that are scattered throughout the section. So these here, okay, these angular atrophy fibers, okay? So these are all type 2 fibers because they are dark, all right? So this is a type 2 atrophy, which is a non-specific finding. All right, the photo on the left-hand side of the slide is an ATPase reaction on this section of muscle done at a pH of 9.4. Let's put pH 9.4. Okay, the grouping of dark type 2 fibers, okay, and light type 1 fibers is diagnostic of a neuropathy. So the photo on the right-hand side of the slide is a muscle stain with the ATPase reaction done at 4.3 pH. So, oh goodness, pH of 4.3. Okay, these results are the reverse of the slide on the left. The dark fibers are type 1 and the light fibers are type 2. The next method is acid phosphatase for muscle biopsies. So this staining technique indicates the presence of inflammatory cells in the muscle biopsy specimen. Acid phosphatase is a marker enzyme for the presence of lysosomes. So muscle fibers in acid maltase deficiency can show an increase in the acid phosphatase level. Acid phosphatases are phosphomonoesterases that hydrolyze esters of orthophosphoric acid at an acidic pH. After that hydrolysis, the alcohol residue of the naphthyl ASBI phosphate is reacted with hexadized pyrarocyanine that produces a highly colored insoluble azo dye. No fixative is used for this technique, and skeletal muscle has an internal control, so neutrophils and histiocytes will stain positively with this stain method. The reagents needed are 2 normal hydrochloric acid, pyrarocyanine hydrochloride, 4% sodium nitrite, Verona acetate buffer, incubating medium, substrate solution, and methyl green. To perform the acid phosphatase stain, frozen sections of unfixed tissue are cut at 10 microns and mounted on slides. The sections need to be incubated in incubating solution at 37 degrees Celsius for 1 hour. After the 1 hour period, rinse the sections in distilled water. After that distilled water rinse, slides should be placed in 2% methyl green for 2 minutes. Then dehydrate sections rapidly at 95% and 100% alcohol. Clear in xylene and then mount with a synthetic resin. When stained properly with the acid phosphatase stain, sites of acid phosphatase activity will be red, where the background will be green. The photo here shows an acid phosphatase reaction used on a frozen section of muscle. This shows a necrotizing or inflammatory myopathy. Acid maltase deficiency will give a positive reaction, and this is something seen in something called Pompe's disease. And Pompe's disease is a genetic condition where glycogen builds up in the lysosomes of the body's cells. This is caused by the lack of a digestive enzyme. This disease results in severe muscle weakness and also wasting of the muscle. So that was acid phosphatase staining method. This is alkaline phosphatase stain for muscle biopsies. So this detects regenerating muscle fibers. Alkaline phosphatase hydrolyzes esters of orthophosphoric acid at an alkaline pH, approximately 8.6 to 8.8. The alcohol residue of the naphthyl ASBI phosphate after enzymatic hydrolysis is reacted with fast red violet to produce a highly colored insoluble azo dye. No fixative is used here, and skeletal muscle has an internal control. This is because alkaline phosphatase is present in small amounts within the blood vessels. Reagents needed for the staining method are 0.2 molar tris, 0.1 molar hydrochloric acid, tris buffer at a pH of 8.74, incubating medium, and either Mayer or Harris hematoxylin. To perform this staining technique, frozen sections of unfixed tissue are cut at 10 microns and mounted on slides. Frozen sections should be immersed in incubating medium and incubated at room temperature for 30 to 60 minutes. Remove the slides, then rinse in distilled water. Then counterstain for 30 seconds to 1 minute in hematoxylin. So this is an optional step. So if the counterstain optional step is used, you'll want to wash sections well with several changes of distilled water, and then of course mount sections with an aqueous mounting medium. When stained properly with the alkaline phosphatase technique, sites of enzyme activity will be pink-red to red in color. Nuclei will be blue. This photo shows a skeletal muscle frozen section that has been stained with this technique. This shows several degenerating muscle fibers. So here's sites of enzyme activity here, because it's that like pinkish red or red color. NADH diapherase is a technique that demonstrates any abnormalities within the mitochondria, Z-band material, and the sarcoplasmic reticulum. In this technique, a tetrazolium salt acts as a hydrogen acceptor. NADH can't reduce this salt directly, so the reduction is attributed to the flavoprotein enzyme, so it's called a diapherase reaction, which means to carry. There is no fixative used for this technique, and skeletal muscle has an internal control. Reagents needed for the NADH diapherase technique are saline solution, phosphate buffer at a pH of 7.4, NBT solution, and incubating medium. To perform the NADH diapherase technique, frozen sections should be air-dried and cut at 10 microns and mounted on slides. Sections should be incubated in the incubating medium at room temperature for 30 minutes. After the 30-minute incubation, slides should be rinsed in distilled water, then mounted with Immumount or glycerin jelly. When properly stained with this technique, sites of enzyme activity will be seen as dark purple deposits. Z-band material, sarcoplasmic reticulum, and mitochondria react very strongly. Type 1 muscle fibers will be dark purple, and type 2 muscle fibers will be light. This photo shows a frozen section of normal skeletal muscle stained using this technique. Type 1 fibers are dark here, and type 2 fibers are light. The photo on the left-hand side of this slide shows a muscle fiber stained with the NADH diapherase technique that shows the central area to be devoid of mitochondria and oxidative enzyme activity. The presence of many of these central cores is usually diagnostic of something called central core disease. The photo on the right shows a frozen section of muscle stained with this technique that reveals many target fibers. These are diagnostic of a neuropathic disease. Succinic dehydrogenase, or SDH, is a technique that is used to further identify the source of NADH diapherase activity. This is because only mitochondria show positive SDH activity. SDH is a mitochondrial enzyme that forms a chain with cytochromes. The enzyme is demonstrated by incubating fresh frozen sections with succinate substrate in the presence of a tetrazolium compound such as NBT. No fixative is used here, and skeletal muscle has an internal control. How nice of the skeletal muscle to have internal controls for these things. Reagents for the SDH technique are 0.2 molar phosphate buffer at a pH of 7.6, 0.2 molar sodium succinate solution, NBT solution, incubating medium, physiological saline, and 10% formalin saline solution. To perform the SDH technique, a frozen section of unfixed tissue is cut at 10 microns and then mounted on the slide. The section should be incubated at 37 degrees Celsius in an incubation medium for 30 minutes. After that 30-minute period, it should be rinsed in physiological saline. After this rinse, fix in 10% formalin saline solution for 10 minutes, and then rinse in 15% alcohol for 5 minutes. Then mount with aqueous mounting medium. When correctly stained with this technique, sites of SDH activity will be blue. The photo on the right-hand side shows a frozen section of normal muscle tissue using the SDH reaction. Type 1 fibers are dark, and type 2 fibers are light here. Only mitochondria show a positive reaction with this technique. The phosphorylase stain for muscle is useful in the diagnosis of McArdle disease. McArdle disease is a genetic disorder that occurs due to the deficiency or absence of an enzyme called myophosphorylase. Patients with this disease have painful muscle cramps, weakness, and fatigue. In vitro, muscle phosphorylase produces an unbranched polysaccharide of amylose type from glucose 1-phosphate. The length of the unbranched chains formed is proportional to the amount of phosphorylase activity present. So when stained with iodine, it will vary in color depending on the length of the chain. If the branching enzyme present in muscle is allowed to act, it will form 1,6 glucosidic linkages, or the branched polysaccharides of glycogen or amylopectin. Alcohol is usually added to the incubation medium to prevent the action of branching enzymes. Glycogen is added to the incubating medium as well as a primer, and insulin is an activator. Once the tissue is ready to stain, then the tissue should be fixed in cold acetone for 5 minutes. Normal skeletal muscle has an internal quality control. Patients that have McArdle disease do not have an internal control in their skeletal muscle tissue. So in this case, a slide from a previous normal skeletal muscle must be used as a control and the test repeated. Reagents needed for the phosphorylase stain are acetate buffer with a pH of 5.9, incubating solution, gram iodine, dilute gram iodine, and an iodine glycerin mounting medium. To perform this procedure, frozen tissue that is unfixed is cut at 10 microns and mounted on slides. Section should be air dried and left for 10 minutes at room temperature. Then the tissue should be fixed in cold acetone for 5 minutes. After that fixation, it should be incubated at 37 degrees Celsius in a capped jar for 1 hour in 15 milliliters filtered stock solution and 3 milliliters of absolute ethyl alcohol. After this 1 hour incubation, shake off any excess medium, wash briefly in 40% alcohol, and then allow to air dry. After it's air dried, fix in absolute alcohol for 20 minutes and then air dry it again. Then stain and dilute gram iodine for 6 to 7 minutes and then mount in glycerin iodine. When properly stained with this phosphorylase technique, any phosphorylase activity will have varying shades of brown, blue, and purple. A total absence of phosphorylase activity indicates that the patient has McArdle disease, which we've discussed before. This photo shows a frozen section of muscle that has been stained using this technique. The color formed when the section is stained with iodine is determined by the length of the unbranched chains formed or the amount of phosphorylase activity present. A negative reaction is yellow. An intense blue-black color displayed by the longest chains. So type 2 fibers usually have the most intense reaction, and darker fibers seen in this section, of course, are type 2 muscle fibers. There are non-enzymatic procedures used for staining for muscle disorders. So these are periodic acid shift, or PAS, oil red O, and the modified Gamori technique. The periodic acid shift, or PAS stain, is used for the detection of glycogen storage diseases. We've already talked about the PAS stain in previous lectures, and the procedure isn't any different for muscle. This photo shows a frozen section of a normal muscle that has been stained with the PAS technique. The normal checkerboard pattern of fiber types can be seen in this photo. The oil red O stain is used for the detection of lipid storage disease. And again, we've already talked about the oil red O stain in previous lectures, and the procedure isn't any different when it's used for staining muscles. This photo shows a frozen section of a normal muscle that has been stained with this technique. The modified Gamori trichrome technique is used for the detection of changes in normal morphology of both muscle fibers and its surrounding connective tissue. This is an excellent stain for striated muscle and peripheral nerves. Architectural alteration in muscle fibers, such as nemeline rods or ragged red fiber changes, are readily seen because of differential staining of pathological inclusions and the sarcoplasm. Proliferation of endomysial connective tissue is also demonstrated with this technique. Tissue should not be fixed, and the stain doesn't require an external control. The reagent needed for this technique is just the Gamori trichrome solution. To perform the modified Gamori trichrome technique for muscle sections, frozen sections that are unfixed should be cut at 10 microns and then mounted on slides. The frozen section should be allowed to air dry at room temperature. Do not fix these, obviously. And then it needs a 5-minute stain in Harris hematoxylin. After this 5-minute stain, wash well in distilled water to remove any excess hematoxylin. Then it needs a 10 to 30 minute stain in trichrome solution. And after that 10 to 30 minute period, it needs to be differentiated by a few dips of 0.2% acetic acid. Then dehydrated in 95% in absolute alcohol, cleared in xylene, and then mounted with a synthetic resin. When properly stained with the modified Gamori trichrome technique, nuclei will be red-purple in color. Myofibrils will be bluish-green, intermyofibril or sarcoplasm, myelinated nerve twigs, nemeline rods, and abnormal mitochondria will be red. Interstitial connective tissue appears light green in color. So this photo shows a frozen section of muscle stained with the modified Gamori trichrome technique. This muscle is stained greenish blue. The photo on the left-hand side of this slide shows a muscle biopsy specimen that's stained using the modified Gamori technique. This biopsy was put in formalin briefly, and this causes the staining pattern to be affected. The photo on the right-hand side of the slide shows a ragged red fiber in the center of this modified Gamori technique stained tissue. This indicates a mitochondrial disease. Okay, so that wraps it up on enzyme histochemistry. If this video helped you, go ahead and give it a like. Please consider subscribing to my channel for more educational laboratory content. Until next time.